In the spring of 1947, in the dry hills along the western shore of the Dead Sea, a Bedouin shepherd searching for a stray goat is said to have thrown a rock into the mouth of a cave cut into the cliffside, expecting nothing more than the dull thud of stone on stone. Instead, according to the story that has been passed down for decades, he heard something break, a hollow, unmistakable crack of pottery shattering somewhere in the dark. When he and his companions eventually worked up the nerve to climb inside, they found tall clay jars, sealed and waiting, and inside some of them, wrapped in aged linen, were rolled sheets of leather covered in ancient handwriting. Nobody involved that day understood exactly what they'd found, but they had just become the first people in nearly 2,000 years to lay eyes on what the world now calls the Dead Sea Scrolls. It's difficult to overstate what those scrolls turned out to be. They include the oldest known copies of the Hebrew Bible ever discovered, texts copied while the Second Temple in Jerusalem still stood, centuries before the versions most people read today had been standardized, with some manuscripts predating the next oldest surviving biblical texts by more than 1,000 years. They survived thanks to an accident of geography. The extreme aridity of the Dead Sea region, the lowest point on Earth's surface, preserved organic material that would have decayed almost anywhere else, turning the desert into an unintentional vault for 20 centuries. It's worth pausing on just how unlikely that preservation actually was. Parchment, being organic animal tissue, is normally one of the more fragile materials archaeologists ever work with, vulnerable to moisture, mold, insects, and the ordinary passage of time in almost any climate on Earth. The specific combination of conditions at Qumran, bone-dry air, stable cave temperatures, and sealed clay storage jars that kept out both moisture and pests, created something close to an ideal natural preservation environment, one that scholars have said would be extraordinarily difficult to replicate anywhere else on the planet. Without that precise convergence of circumstances, it's entirely possible this entire body of material would have disintegrated into dust centuries before anyone had the chance to read a single word of it. What followed that initial discovery was a slow-building wave of exploration. Between 1947 and 1956, searchers combed 11 caves near the ancient ruin of Qumran, and archaeologists, explorers, and local Bedouin uncovered an extraordinary quantity of material. But here is where the romance of discovery runs into the sheer difficulty of what came after. The scrolls were not, for the most part, intact documents. They survive as roughly 25,000 fragments, some large enough to read comfortably, many no bigger than a fingernail, believed to represent something like 1,000 separate original manuscripts, all mixed together with no titles, no page numbers, and no way to know at a glance which fragment belonged with which document. For close to 70 years, the primary tool available to reconstruct this fractured library was the trained human eye. Scholars learned to recognize individual scribal hands, the particular slant of a letter or the curl of a stroke, and studied the physical characteristics of the parchment itself, its color, thickness, and pattern of wear, reasoning that fragments sharing a scribal hand and a similar physical appearance likely came from the same original scroll. It was careful, expert work, but it carried an inherent and acknowledged risk. Two scribes trained in the same school could write in nearly identical styles, and two unrelated pieces of leather could age in similar ways, meaning a wrong match could stitch together a sentence, or even a whole passage, that no ancient scribe had actually written. To grasp the scale of the challenge scholars faced, it helps to imagine emptying out roughly 1,000 different jigsaw puzzles onto a single table, with most of the pieces from each puzzle missing entirely, no box lids to show the finished picture, and no way to tell at a glance which scattered piece belonged to which puzzle in the first place. Many of the surviving pieces looked nearly identical in color and texture, the same brown, brittle leather and faded ink, with nothing but the subtlest variations in handwriting to distinguish one original document from another. That was the task facing generations of scholars. Not simply translating ancient text, but first reconstructing which physical scraps of 2,000-year-old animal skin had ever belonged together in the first place, a job that had to be done before serious textual interpretation could even begin. Out of decades of that careful, if inherently uncertain, work came a dominant theory about who produced the scrolls and why they ended up hidden in those caves. Scholars pointed to the Essenes, a small, ascetic Jewish sect described in ancient sources as having withdrawn from mainstream society to live in disciplined isolation near the Dead Sea, at a settlement called Qumran situated just a short walk from where the first scrolls were found. Some of the texts themselves seem to support the connection. Community rule books governing shared property and behavior, solar calendars diverging from the lunar calendar used by religious authorities in Jerusalem, and sharp critiques of the temple priesthood, the language of a group convinced it alone preserved the authentic faith. According to the standard account, when Roman legions advanced through the region in 68 CE to crush a Jewish revolt, the Qumran community hid its library of scrolls in the surrounding cliffs, hoping to retrieve them once the danger passed. They never returned, and the scrolls waited in the dark for 19 centuries. It was a tidy, emotionally satisfying theory, and for a long time it held up reasonably well. But it also had loose threads that never fully resolved. Scholars examining the biblical texts among the scrolls found genuine variation between copies of the same book, differing wording, rearranged passages, and material present in one copy but absent from another, differences too substantial to be simple copying errors. That raised an uncomfortable question. Why would a single, insular sect convinced of its own doctrinal purity preserve multiple, sometimes contradictory versions of its own scripture, including versions that seemed to reflect the very religious perspectives it had supposedly rejected? The physical parchment itself was inconsistent too, ranging from finely prepared, even skins to rougher, more crudely processed ones, a level of variation that seemed unusual for material produced by a single, self-contained community. And a handful of scrolls appeared to have been written before the Qumran settlement itself was even established, raising the odd possibility that the supposed source of the library predated its own founding. Scholars offered reasonable explanations for each of these problems individually, but the explanations had the feel of patches over cracks that never fully disappeared. For decades, there was no way to look past those cracks, because every available method depended on studying what could be seen with the eye: ink, handwriting, and the surface appearance of leather. The deeper question of where each individual scrap of parchment had actually originated seemed permanently out of reach. That changed, unexpectedly, through a conversation between two researchers at Tel Aviv University who had almost nothing in common professionally. In 2012, Oded Richavi, a molecular biologist who studies genetics in microscopic roundworms, and Noam Mizrahi, a scholar of ancient Hebrew literature, ended up seated next to each other on a bus during an orientation program for new faculty. What began as small talk about their very different fields eventually turned into a genuine collaboration, built around a simple, almost obvious question that 70 years of scholarship had never made central to the puzzle. What were the scrolls actually made of? The answer was familiar to everyone in the field. Animal skin, parchment prepared from the hides of sheep, goats, or occasionally cattle, the standard writing surface of the ancient world. But to a geneticist, that familiar fact suggested something nobody had previously pursued. If the scrolls were made from animal skin, then they were made from animals, and animals have cells, and cells carry DNA. The parchment itself, beyond being a passive surface for ink, was really a second separate document, one written in the language of the genome rather than in a scribe's handwriting, a record of a specific animal's ancestry embedded in the material the scribes had written on. Actually pursuing that idea, though, ran into an immediate and serious obstacle. The Dead Sea Scrolls are among the most carefully guarded artifacts in the world, sacred to multiple religious traditions and extraordinarily fragile after 2,000 years in the caves followed by decades of handling by researchers. Conventional DNA extraction typically requires cutting or drilling into a sample, destroying a piece of the object in the process, and there was never any realistic possibility that the Israel Antiquities Authority, the scroll's custodian, would permit researchers to remove and destroy fragments of biblical text to satisfy a genetic study. The team needed to sample the animal material without sampling the actual scroll fragments themselves, a condition that initially looked close to a contradiction. The eventual solution came from something almost embarrassingly mundane: the fine parchment dust that had, over decades of careful handling, photography, and storage, settled at the bottom of the sealed containers holding the fragments. Every time a fragment was lifted or examined, however gently, microscopic particles of the leather itself flaked loose and drifted down into the container. That dust wasn't waste. It was the same collagen, the same 2,000-year-old tissue as the fragments above it, just broken down past the point of being legible, and it carried the same genetic information. Researchers were also able to draw minute samples directly from blank margins and already damaged edges of a small number of fragments, where a few grains of material could be collected without touching or endangering any surviving text. Between 2016 and 2020, the team ultimately worked with genetic material recovered from 26 different scroll fragments, along with other leather items connected to the Qumran caves, including sandals, a garment, and water skins, gathered without ever cutting into or damaging a single piece of inscribed parchment. Sampling the material was only the first hurdle. The DNA itself presented a much harder problem, because it had been through an ordeal that destroys genetic material almost completely under normal circumstances. The parchment-making process itself, the scraping, stretching, and chemical treatment of raw hide, degraded the tissue before a single word was ever written. 2,000 years in the caves, subject to heat and the slow chemistry of decay, fragmented what DNA remained into short, badly damaged pieces. And 70 additional years of human handling layered modern contamination directly on top of whatever ancient signal survived, since every touch, even through gloves, deposits some trace of the handler's own DNA. To have any real chance of recovering a usable signal, the research team, working across labs including Uppsala University in Sweden under geneticist Matthias Jakobsson and with additional collaboration from Christopher Mason at Weill Cornell Medicine, had to work under extraordinarily strict anti-contamination protocols, the kind normally reserved for the most demanding ancient DNA research anywhere. Researchers worked in full protective gear inside positive pressure cleanrooms designed to push airborne particles outward rather than allowing anything to drift in, with every surface, tool, and vial treated as a potential contamination risk until proven otherwise. It's worth appreciating just how demanding that standard actually is in practice. Ancient DNA research of any kind is notoriously vulnerable to contamination from modern sources, since a single stray skin cell or an unguarded breath can introduce enough contemporary genetic material to overwhelm a faint, degraded ancient signal entirely. That risk is compounded enormously with material that has already passed through decades of handling by researchers and curators, many of whom, in the earlier history of scroll scholarship, worked with the fragments using bare hands rather than the gloves and protective equipment that later became standard practice. Every one of those historical handling events left behind trace amounts of modern human DNA layered directly on top of whatever ancient genetic material the parchment itself still held, meaning the research team had to develop methods sensitive enough to isolate a genuinely ancient signal from underneath decades of accumulated modern contamination, a technical challenge nearly as difficult as recovering the degraded ancient DNA itself. Within that controlled environment, the team looked for two different categories of genetic material. Mitochondrial DNA, present in hundreds or thousands of copies per cell, was more likely to survive in recoverable quantities even in badly degraded samples, and it could reliably identify which species, sheep, goat, or cow, a given fragment came from. Nuclear DNA, far scarcer and more fragile, offered a much finer level of detail, potentially distinguishing one individual animal from another closely related one, but recovering it reliably from material this degraded required developing new experimental and computational methods specifically for this project. When the results came back, the first finding matched expectations closely. The overwhelming majority of sampled fragments were made from sheepskin, unsurprising given that sheep and goats were the practical livestock of a desert environment, hardy animals capable of surviving on sparse vegetation and limited water. If any scholar had been asked before the sequencing what material the scrolls were likely written on, sheepskin would have been the reasonable guess, and for most of the fragments, that guess held up. But not all of them. Among the sampled fragments, two came back not as sheep, but as cattle, and that single result carried enormous implications. Cattle require abundant water and grazing land, resources that simply don't exist in the arid cliffs above the Dead Sea. Raising cattle at Qumran, let alone producing cowhide parchment there in meaningful quantity, would have been effectively impossible. That meant the cowhide fragments had almost certainly been produced somewhere else entirely, somewhere with rivers and pasture, and had been carried into the Qumran caves from outside. That finding pressed directly against a central assumption of the traditional theory. If the scrolls represented the output of a single, isolated community working in one place, they should share a single point of origin. The cowhide fragments suggested otherwise, indicating that the material gathered in those caves came from more than one source. The clearest illustration of what this meant came from the book of Jeremiah, which survives among the scrolls in two notably different versions. One longer and closer to the traditional Hebrew Bible, the other shorter, with material rearranged or missing. Scholars had debated for years whether specific fragments from these differing versions might actually belong to the same original scroll. The genetic analysis settled at least one such case definitively. Two fragments long assumed to belong together turned out to come from entirely different animals, one from sheep, the other from cattle, meaning they could not have been part of the same document at all, and in fact represented two genuinely separate textual traditions of Jeremiah that had arrived at Qumran from different directions. A similar pattern showed up in other groups of fragments as well, including copies of the liturgical text known as the Songs of the Sabbath Sacrifice, where genetic analysis indicated that copies found together at Qumran had actually been produced in different places and brought together later, rather than all originating from a single local scriptorium. Kinship analysis of the recovered nuclear DNA also allowed researchers to identify fragments that had come from the same or closely related animals, offering a genuinely new, physical method for grouping fragments that no amount of handwriting analysis alone could provide. Taken together, this evidence pointed toward a picture considerably more complicated than the traditional single-sect theory. Rather than the private archive of one isolated community, the caves at Qumran increasingly looked like something closer to a geniza, the traditional Jewish practice of storing worn-out or textually problematic sacred writings that were too holy to simply discard. Jerusalem lies only about 20 miles from Qumran, and it's entirely plausible that as the Roman assault on the city intensified, people fleeing the destruction carried scrolls out into the desert for safekeeping from multiple communities and origins, not just from a single local sect, hiding them together in the same caves. That reframing carries real historical weight beyond simply settling an old academic dispute. It suggests that in the final generations before the Second Temple's destruction, the biblical text itself was not yet a single, fixed, standardized document, but a living, still evolving tradition, copied in multiple forms by different communities who had not yet settled every question of what counted as authoritative scripture. That's the world into which the earliest Christian communities eventually emerged, a religious landscape considerably more diverse and unsettled than the popular image of one static, ancient library sitting behind Qumran's walls would suggest. It's worth being clear about the limits of what this genetic research can and can't tell us. The study, led by Richavi and Mizrahi and published through Tel Aviv University's collaboration with international partners in 2020, worked with a relatively small sample size, 26 fragments out of the roughly 25,000 that survive, meaning it offers targeted insight into specific, previously disputed cases rather than a wholesale rewriting of everything scholars believe about the scrolls. The traditional scholarship built on decades of handwriting and paleographic analysis wasn't wrong to rely on the tools available at the time, and the genetic findings don't discredit that work so much as add an entirely new layer of evidence on top of it, one written not in ink but in the biological material the ink happened to sit on. What the DNA offers is a form of confirmation, an occasional correction, that handwriting alone could never provide. An objective, physical record of where a given scrap of 2,000-year-old skin actually came from, hiding in plain sight in the very material scholars had been studying for 70 years without ever thinking to ask what it was actually made of. There's a broader lesson in this story that extends well past the Dead Sea Scrolls themselves, one about how different fields of expertise can miss things that are obvious in hindsight simply because nobody working in a given discipline happened to think to ask a question from outside its usual boundaries. Textual scholars had spent generations becoming extraordinarily skilled at reading ink, handwriting, and the visible surface of ancient parchment, and geneticists had spent decades developing increasingly sophisticated tools for reading degraded ancient tissue from bones, teeth, and other biological remains. Neither group had much reason, on their own, to think of 2,000-year-old writing material as a biological specimen in its own right, worth sequencing independently of the text written on top of it. It took a chance conversation between two people from entirely separate departments, sharing nothing more than a bus ride during a faculty orientation, to notice a question sitting in plain view that an entire field had somehow never made central to its work. That kind of interdisciplinary accident, more than any single finding about sheep or cattle or the book of Jeremiah, may be the most instructive part of the whole story, a reminder that some of the most significant discoveries come not from new instruments alone, but from someone finally asking an old object what, exactly, it was made of.